Hello everyone, I'm a junior student from Kunshan University. Today, I would like to introduce Plasmid in bacteria, how we conducted a relevant lab experiment in Biology 110, and its connections to real-world applications. First, please allow me to introduce Plasmid. The word plasmid was coined by Joshua Lederberg in 1952. Plasmid is now defined as a small, circular, double-stranded DNA molecule that carries accessory genes separating from those of a bacterial chromosome. Compared to normal chromosome, it usually carries fewer genetic information, containing around 10,000 base pairs of DNA. Although plasmid is common in bacteria, other organisms, like yeast, which is eukaryote, also have plasmid. Following a deeper understanding of plasmid is its applications. Plasmid has been used in recombinant DNA technology in biogenetics since 1970s. The picture on the right is the image of DNA of the PSC101 plasmid observed by electron microscope. The plasmid has been used for the first successful DNA colony experiment. Then I should introduce the structure of plasmid. There are three important components in a plasmid. The first is origin of replication. It initiates the replications of plasmid. Multiple colony site is also essential. It allows different restriction enzymes to cut the plasmid to insert possible new genetic information. To transcribe a messenger RNA from the plasmid, an enzyme, RNA polymerase, is necessary for the synthesis of RNA. The promoter, therefore, is necessary as a site for the binding of RNA polymerase. For some plasmid, they have special genetic sequences giving them different abilities. For example, plasmid with F factor allows the bacteria to form the mating bridge pili with another one. Through pili, it can transfer its DNA to others through conjugation. Plasmid with R factor gives the bacteria resistance to antibiotics, which makes treatment of bacterial infection more difficult. As a source of foreign and genetic information, plasmid is very important for bacterial transformation. In bacterial transformation, one bacterium can take the foreign DNA from its surroundings. Through exchange of homologous DNA segments, the recipient bacterium can integrate the foreign DNA into its own chromosome. Through transformation, the transformed bacteria now have genetic information from two different cells. The transformation often gives an advantage to bacterial survival. For example, it can develop antibiotic resistance and allow its survival under the influence of many antibiotics. Plasmid with F factor also plays an integral role in bacterial conjugation. Only when one bacterium contains plasmid with F factor can it have conjugation with other bacteria. Different from transformation, the recipient bacteria does not take foreign DNA from surroundings, but from another bacteria. Accompanied by replication of plasmid, the donor bacterium injects the plasmid into the recipient bacterium through pili. After plasmid transfer, the recipient bacterium also has one plasmid with F factor. Therefore, it becomes a new donor bacterium and can donate its plasmid to others. In our fifth lab experiment, we conducted an experiment to transform E. coli by using PUC19 as the plasmid. The plasmid contains both AMPR gene and the LACZ gene. According to our expectations, the transformed E. coli should develop resistance to ampicillin, one type of antibiotics, and therefore they can survive under the environment of ampicillin. However, as the color of E. coli is off-white, how can we know whether there are bacteria surviving or not? Therefore, we use IPTG to detect the existence of transformed bacteria in our plate. The LACZ gene will synthesize one enzyme, which will hydrolyze the IPTG. The hydrolyzed IPTG product will show blue, indicating the existence of transformed bacteria. In our lab, we first added PUC19 plasmid into the tube containing E. coli. We then gave the E. coli a heat shock to trigger the transformation process. After overnight incubation, we check our experiment results. The imager on the left shows our results. We can see that the E. coli grew quite well on the plates without ampicillin. the ones at the up and the bottom left, and formed a bacterial lawn. For the plates with ampicillin, however, only the plate that was supposed to contain transformed E. coli, the one at the up left, showed blue colonies. For untransformed E. coli, they cannot survive in the environment of ampicillin. The results show that some, but not all the E. coli successfully took the foreign DNA and developed resistance to ampicillin. Compared to the E. coli on the plate without ampicillin, we can see that a gap exists in the concentration of the bacteria, which is influenced by transformation rate. The transformation rate is controlled by multiple factors, including the plasmid side, the environmental pH and temperature. Many scientists are now exploring more factors to regulate the transformation rate of bacterial transformation. So, what is the importance of plasmid? Why should we care about it? Well, one thing that you should know about plasmid is that it contributes to the prevalence of antibiotic resistance. Antibiotic resistance is one form of antimicrobial resistance. It means that bacteria develop the ability to defeat the drugs designed to kill them. For bacteria, such drugs are usually antibiotics. How can plasmid give bacteria the ability to survive antibiotics? It is related to transcription and translation process in bacteria. In bacteria, DNA is transcribed into messenger RNA. Messenger RNA, in turn, will be translated to synthesize the proteins. The proteins act as enzymes and function usually to protect bacteria from antibiotics. There are three common mechanisms. The first one is that the plasmid allows the production of bacteria enzymes to hydrolyze antibiotics. The plasmid can also reprogram the antibiotic targets, usually some membrane structure, which makes antibiotics unable to detect the bacteria. Therefore, bacteria can escape antibiotics. The last mechanism is that some transformed bacteria can pop up antibiotics through its membranes and keep the level of antibiotics under the threshold of toxicity. What are the causes of such resistance? The root cause is overuse of antibiotics. The overuse of antibiotics filters the bacteria and only leaves the bacteria with resistance to antibiotics, which is also part of evolution selection. Therefore, for the bacteria that survive, most of them now have resistance to antibiotics. Some places, like hospitals, also exposes bacteria to environment with a lot of plasmids. Through transformation, it also produces more bacteria with antibiotic resistance. As we have mentioned before, the plasmid that carries genes for antibiotic resistance is R plasmid, where R means resistance. Many R plasmids, however, also contain F factor. Therefore, they can transfer their antibiotic resistance genes horizontally between different bacteria through conjugation. As time passes by, some bacteria have developed resistance to several antibiotics and become superbugs, which are very dangerous and life-threatening. To avoid the spread of antibiotic resistance and preserve the effectiveness of newly developed antibiotics, antimicrobial stewardship is launched to monitor the use of antibiotics in healthcare and agriculture, preventing its overuse. After learning the role of plasmid in antibiotic resistance, are you now afraid of plasmid and considering it as the bad guy? Well, with appropriate usage, plasmid is also a powerful tool in biogenetics to benefit people. For example, plasmid has been used to produce human insulin for over 4 decades. Human insulin is a protein with a simple structure of 51 amino acids on A and B chains. In 1978, the scientist team shown on the right picture inserted special plasmid into E. coli. The genetic information on the plasmid allows the E. coli to produce human insulin, which saves millions of lives by treating diabetes type 1. Plasmid is also promising in producing vaccines. Many scientists are now devoted to modifying bacteria to produce 1 or 2 pathogen proteins. After injecting the transformed bacteria, the produced pathogen proteins will activate the B and T immune cells and allows the production of memory cell, which offers immunity. Compared to traditional vaccines, it is safer, more stable for transportation, and more cost-effective for a large-scale production. At the end of my presentation, I would like to express my gratitude. First of all, I would like to thank Professor Lee, my professor in Biology 110, and my classmates. They helped me a lot by helping me understand the concept and solve a lot of my questions. I also would like to thank my parents, who listened to my presentation and gave me valuable feedbacks. Last but not least, I would like to thank my cats, who offer me emotional support and relieve my stress. The left one is Peanut and the right one is Curry. I love them and thank them for their company, although they probably never know what a plasmid is. And here are my references. I think that's all for my presentation. Thank you for your listening and time. If you have any question, please feel free to email me.